So today we go for a particular class regarding your paper 604. The topic is monoclonal antibody. So have you seen the particular slide? Is the screen is visible?
Yes, sir.
Okay.
Yes, sir.
Okay. So the topic is monoclonal antibody. Okay. Okay. So in this particular topic, we basically go for the different aspects. So these aspects are basically number one, what is antibody? Okay. First we will discuss about what is the different types of antibodies are there. Then you go for what is a monoclonal antibody. Then the history, how the monoclonal antibodies are, they are discovered by different scientists. And then production of, or maybe the process of monoclonal antibody, how the monoclonal antibodies are produced by different mechanisms. And then last one, the application of the monoclonal antibody. Okay, so you all know the antibodies basically they are derived from a particular cell which is called as a white blood cell. Okay, so white blood cells, basically the lymphocytes they can produce the antibodies. Okay, so in this particular diagram you can see, so this is a one type of antibody. Okay, so in the antibody they having different types of sites, and these particular sites are called the antigen binding site. Okay, so in one antibody two different antigen binding sites are there. Okay, so in this antigen binding sites, what happen, all the antigens of different foreign particles, it may be bacteria, maybe virus, okay, or maybe some other molecules. So these antigens are binding in this particular area. Okay, that means this antibody is specific for a particular antigen. Okay, because this particular site have to be matched with the antigen. Okay, so suppose this is one antigen, this antigen is matched by this particular antibody, and this antibody is secreted by a particular cell, and this cell is called plasma cell. Okay, so plasma cell is a one type of cell which is derived from the lymphocytes. Okay, so one type of B lymphocyte is there. So one type of B lymphocyte which is transformed into the plasma cell, and the plasma cells can secrete the antibodies. Okay, and these antibodies can they can identify or recognize a special type of antigen. Okay, suppose you can heard about a recent topic regarding the SARS-CoV-2. Okay, so there is a particular protein present in the virus outer membrane. The protein is called spike protein. Okay, so for this particular spike protein, different types of antibodies are generated. Okay, so we already given our doses just like vaccination. So when we are vaccinated, that means our body becomes immunized for the particular spike protein. Okay, and that's why our body can generate the antibodies for the particular spike protein which is present in the particular virus. Okay, so now our body can recognize any type of spike protein which is delivered by a particular SARS-CoV-2 virus. Okay, so just like that, these antibodies are responsible to identify different types of antigens which are actually the foreign particle, not self particle. These are all our foreign particles. Antigens are basically all our foreign particles. Based on that, our antibodies are developed and they can recognize the antigens. Okay, so this is about the basic knowledge about the antibody. Okay, now what is an antibody? Okay, how their looks like and what is the different structure of an antibody. Okay, so you can see this is a definite structure of an antibody. This is a definite structure. So in a particular structure of an antibody, you can see two different chains are there. So this is a long chain. You can see starting from here, the blue color, starting from here up to that much. Okay, this is a long chain. This is also another long chain, and these two are the small chain. Okay, so they having two smaller chain and two long chain. Okay, and these two long chains are called as the heavy chain. So these two are the heavy chain. Okay, and these two are the light chain. This is one light, this is another light chain, and these two are the heavy chain. So in an antibody, basically two different heavy chains are there and two light chains are there. Okay, then you see over here it is written as VL. You can see VL, VH, VH, VL. Okay, now what is V? V means actually this is the meaning of variable. Okay, V means variable, and over here it is written as C. C means constant. Over here it is written as C. C means constant, V means variable. Okay, so you can see over here this is called as a VL. That means variable region of light chain, and this is VH, variable region of heavy chain. This is also variable region of heavy chain. This is also variable region of light chain. Okay, and below you can see CL, CH, constant region of light chain, constant region of heavy chain, constant region of heavy chain, constant region of light chain. Okay, so in a light chain, two different areas are there. One area is variable, one area is constant. In the heavy chain also, one area is variable and one area is a constant. Okay, so why this particular area is called variable region? Why this area is called a variable region? Because you can see in this area the antigen binding site is there. All the antigens, they can bind over here. Okay, and this antibody is specific for a particular antigen. Okay, this antibody is specific for a particular antigen. So why this is called variable? Because if the antigen structure is somewhat variable, suppose the antigenic structure also, they are also variable because of the particular organism. It may be bacteria or maybe virus. Okay, when they getting inside a human body, okay, they just try to change their structure. Okay, they have to remain alive in the body condition, in the human body condition. That's why they change their structure. So when they change their structure, their antigenic structure also changes. Okay, so based on the antigenic structure, this antibody, they can also change their structure so that this region can bind to the antigen perfectly. Okay, that's why this region is called variable region. So this region is changes depending upon the changes nature of the antigen. Okay, so that's why this area is called variable region. This is variable, but these regions are called constant. They remain same. Their structure remains same. So this long chain also having the same region. You can see constant region heavy chain, constant region heavy chain, constant region heavy chain, constant region heavy chain. Okay, so these are all constant region. Only this part are called variable regions. Okay, now see this particular structure is bounded by different types of bridges. Okay, you can see. So these are some bridges are there. This is one bridge, this is another bridge, this is another bridge. So these bridges are called disulfide bridges. Okay, what it is written as S and S, that means these are disulfide bridges. This here also disulfide bridge, here also disulfide linkage is there. So based on the disulfide linkage, both the heavy chain and the light chain, they are attached to each other. Okay, so question is that why they having the disulfide linkage? Okay, because during the formation of the protein, because antibody is what? Antibody is nothing but this is a one type of protein. So protein having the different types of polypeptide chains. Okay, so this is a long polypeptide chain which is called heavy chain, and this is a short polypeptide chain which is called light chain. Okay, so during the production of the protein, the light chain and the heavy chain, they are formed in different areas. Okay, different cell structure, light chain is formed and the heavy chain is formed. Okay, and then this heavy chain and the light chain both are accumulated inside the endoplasmic reticulum and then form a new type of structure, and this new type of structure is called antibody. That means the light chain and the heavy chain, they are generated in different parts of the body. Actually inside a cell, different areas they are produced, and finally they are unite together to form a definite antibody. So during the union of heavy chain and the light chain, these disulfide linkage are there. They can bind this chain into this chain to form a complete structure of the antibody. Okay, so basically this is the
accumulation of heavy chain and light chain ever repeat koribo kotha.
Okay, now see okay, so now see inside a cell we having a nucleus. So nucleus having the DNA. So DNA can generate the mRNA. Okay, and this mRNA undergo protein synthesis and they can produce the polypeptide chains. Okay, now see in a DNA different genes are there. Gene for coding for the heavy chain, gene for coding for a light chain, these are different. Okay, gene for coding for heavy chain and the gene for coding for light chain, these are different. Two different separate genes are here. Suppose gene A can code for heavy chain, gene B can code for light chain. Okay, then both the genes, they can produce two different mRNAs. Okay, now both the mRNAs getting inside different types of ribosomes, and from one ribosome they produce light chain polypeptide chain, and from this mRNA they can produce the heavy chain polypeptide chain. Okay, then this heavy chain polypeptide chain and the light chain polypeptide chain both are getting inside the endoplasmic reticulum. So when they getting inside the endoplasmic reticulum, both the proteins are modified. This is called post-transcriptional modification. Based on this modification, they produce a new type of protein, and in this protein, this light chain and the heavy chain both are joined together, okay, by a linkage, and this linkage is called disulfide linkage. Okay, based on this disulfide linkage, both the light chain and the heavy chain, they are joined together. Okay, this is one type of post-transcriptional modification, and this is occurring inside the endoplasmic reticulum. Now understood, Indaksi?
Yes, sir.
Okay.
Thank you.
Okay. So this is about the basic structure of an antibody. Okay, this is about the basic structure of an antibody. Now, basically, in this chapter we discuss about the monoclonal antibodies. Okay, so we do not go for details about the antibody. Just try to remember what is the basic structure of antibody and what are the different types of antibodies. Okay, in the next class we can go for the immunoglobulins. Okay, then we can basically identify the detailed structure of the antibodies. Okay, so in this class we only go for the basic knowledge about the antibody. Okay, so this is the structure of the antibody. Now, in a human body, antibodies are 5 different types. 5 different classes of antibodies are there. Okay, so these are the 5 classes. You can see IgG, IgE, IgD, IgM, and IgA. Okay, so immunoglobulin G, immunoglobulin E, immunoglobulin D, immunoglobulin M, and immunoglobulin A. So these are the 5 classes of antibodies found in the human body. Okay, among these 5 classes, these 3 classes are exactly similar. You can see IgG, IgE, and IgD. They having the exactly similar structure. All having the Y-shaped structure, and they having the antigen binding region. 2 antigen binding region both sides. IgG, IgE, and the IgD. All having the same structure. But the IgM and the IgA, these are looks like somewhat different. Okay, you can see in the IgA, 2 different antibodies are joined together. Okay, this is called joining chain. This is called joining chain. So based on the joining chain, IgA molecule is formed by union of 2 different antibodies. 2 antibodies are joined together to produce the IgA. Okay, so in the IgA, 4 different antigenic sites are there. 4. 1, 2, 3, 4. 4 different antigenic sites are there in the IgA. Then what is IgM? So in the IgM you can see 5. 1, 2, 3, 4, 5. So 5 different antibodies are unite together in the IgM. That's why IgM is also called as pentameric. This is also called as a pentameric antibody. Pentameric antibody, because 5 different antibodies are joining together. 5 different. Okay, and all are joined by this joining chain. Okay, so this is the basic structure and basic knowledge about the antibody. Okay, just try to remember this is the structure of the antibody, and antibodies are 5 different types found in the human body. But basically in this chapter we are discussing about the monoclonal antibody. Okay, monoclonal antibody. Okay, it is a one type of antibody which is called monoclonal. Now question is that why it is called as a monoclonal. Okay, now we go for details about the monoclonal antibody. Okay, you can see what is a monoclonal antibody. Okay, you can see a monoclonal antibody is an antibody made by cloning a unique white blood cell. The cloning of a unique white blood cell is called as a monoclonal antibody. That means only one type of white blood cells which can generate a simple type of antibody which is called monoclonal antibody. Okay, now see, monoclonal antibody cannot be produced by human body by its own. Suppose we are infected by a virus or we are infected by a bacteria. Suppose I am infected by a virus. So in a virus, different types of antigens are there. Suppose they having the spike protein. Suppose they having some other types of proteins. So different types of proteins are there in the viral body. So when this virus getting inside the human body, what happened? All the different parts of the virus, they are just like one antigen. Okay, spike protein is a one type of antigen, and another protein is a one type of another antigen. Okay, now every antigen can produce different types of antibodies. Okay, every antigen can produce different types of antibodies. That's why our human body, by our natural immunity, we cannot produce monoclonal antibody. We cannot produce because a lots of different antigens are there in a particular virus or in a particular bacteria. That's why the antibodies which are produced by natural immunity, it is called as the polyclonal antibody. Okay, we are generating the polyclonal antibody. We cannot generate the monoclonal antibody because when a virus is attacked, when a bacteria attack us, the virus and the bacteria having poly type of antigens, and poly antigens can produce poly type of antibodies. That's why these antibodies are called poly. This is not called as a monoclonal antibody. This is called polyclonal antibody. Okay. Now, how the monoclonal antibodies are generated by a specific type of white blood cell? How? So this is a one type of artificial insemination. What is an artificial insemination? That means in this condition, we have to injected one type of antigen. We have to inject only one type of antigen, not many type. We have to inject only one type of antigen in the body. Then this injected antigen can generate only one type of antibody. This injected antigen can produce only one type of antibody. Okay, and this antibody, which is produced by a special type of white blood cell, is called as the monoclonal antibody. Okay, so this is a one type of artificial mechanism. It is not a natural mechanism. Okay, naturally we cannot produce a monoclonal antibody, but some cases, in some time, we can produce the monoclonal antibody by natural process. Okay, this is the exception. And what is the exception? This is in case of a particular cancer, a cancer which is called myeloid cancer. Okay, multigenic myeloid cancer. Okay, in case of multigenic myeloid cancer, so this is one type of cancer producing in the plasma cells. This is one type of cancer producing in the plasma cells. Okay, so these plasma cells which having cancerous plasma cells, they can produce monoclonal antibody during the cancer development. Okay, and this is called natural producing monoclonal antibody by cancer cell, which is called multigenic myeloid cancer. Okay, but basically our body cannot produce monoclonal antibody. It is only produced by artificial method. Okay, they can see monoclonal antibodies can have monovalent. I already said only one valent is there, monovalent affinity, binding only to the same epitope. That means they can bind only a specific type of epitope. Only one epitope they can bind. In contrast, you can see in contrast, polyclonal antibodies bind to multiple epitopes. Okay, so polyclonal antibodies are binding to several types of epitopes. Okay, I already said bacteria having different types of epitopes, and our body can produce different types of antibodies for all the epitopes. Okay, and finally the antibody which is produced by natural immunity, this is called polyclonal antibody. Okay, now see another exception. This is called bispecific monoclonal antibody. Sometimes we can also produce a bispecific monoclonal antibody. That means this antibody can recognize two epitopes, two different epitopes they can recognize by the same monoclonal antibody. Okay, this is called as the bispecific monoclonal antibody. This is a one type of exception. Basically, all the monoclonal antibody, they can only recognize only one epitope. Not all, only one epitope they can recognize. Okay, now you can see this is a diagram. So this is the diagram of this is a virus. This virus is called SARS-CoV-2 virus. Okay, so in this virus, you can see this is the red color or pink color area, this is the spike protein. This is a spike protein. Okay, so we all know, you all heard about that. Nowadays, a particular cocktail of monoclonal antibodies are used. Okay, for the what? For the different types of searching for this type of spike protein. Okay, so these spike proteins are basically, they are generated based on this particular antibodies. So someone have unmute your microphone. Please mute yourself. Okay, please mute the microphone. Okay, now see. So this is the particular virus. So this virus having this type of epitopes, these are called spike protein. Okay, so this is only one type of epitope, or only one type of antigen. This is only one type of antigen. Okay, so for this particular antigen, we can generate a one type of monoclonal antibody. Okay, so these are the monoclonal antibody, they are generated. Okay, they are generated and they can bind to the spike protein directly. Okay, they can bind. And when these spike proteins are bind to the monoclonal antibody, then this virus cannot bind to the human cell receptors, because these are the receptors, spike proteins are the receptors, and these receptors are blocked by the monoclonal antibodies. These antibodies can block the spike protein. That's why this virus cannot get inside a human cell. Okay, so this is the one type of mechanism, and this mechanism is done by a recent study which is called the cocktail of monoclonal antibodies are used for the prevention of the particular virus. Okay, so in this condition,
Sir, vaccination of pasot ki monoclonal antibodies form hobo na polyclonal, sir?
Polyclonal hobo. This is not monoclonal. But it is so because you can see we having different types of vaccines. Okay, you can see we are having the Covishield, we are having the Covaxin. Okay, so in case of the Covishield, so we are using only the spike protein. Okay, in case of the Covishield, we are only using only the spike protein. That means during the production of antibodies, by applying the Covishield type of vaccination, then we can produce only monoclonal antibodies for the spike protein only. Okay, but in case of Covaxin, we are using the whole virus. Whole virus we are using for the formation of the Covaxin. So in this condition, our human body can produce polyclonal antibody for the particular virus, because virus having the different types of site. They having the spike protein, they having the other proteins. Okay, so for all the proteins, the Covaxin can generate different types of antibodies. This is called polyclonal. But in case of Covishield, they generate only the monoclonal antibody, only specific for the spike protein.
Sir, to kuntu bisi bhal hoy, monoclonal form hole bhal na polyclonal form hole bhal? Okay,
now see. One question is that suppose this particular virus, okay, this particular virus cannot be dangerous for the human body if they cannot enter through the cell. Okay, if this particular virus, if we can block the entry of this virus inside the cell, that means we can prevent ourselves from the severe disease. Okay, how we can prevent? That means if we have blocked this spike protein, if you can able to block this spike protein, then this virus cannot able to enter inside the human body, or actually not inside the human cell. They cannot enter. Okay, so if these spike proteins are blocked by this particular antibodies, then this virus cannot get inside the human body. Okay, that means polyclonal or monoclonal antibodies, they both are equivalently working for the particular virus. Okay, both are equivalent. Now see, in case of the Covaxin, what happened? They can generate the spike protein antibody. They can also generate the other protein antibodies. So in this condition, you can say that they are most beneficial, because if the spike proteins are not blocked by the particular antibody, then other proteins may be blocked. So in this condition also, we can restrict the entry of virus inside the human body. Okay, but in terms of vaccination purpose, both the vaccines are equally equivalent. Okay, Covaxin as well as the Covishield.
And sir,
what?
If spike vaccine spike protein or mutation ho jay sir, titia to monoclonal antibody to aru kaam kobo nwaribo?
Okay, it is what during vaccination. Okay, during vaccination we are using a particular virus. Suppose we are in a normal SARS-CoV-2 virus during vaccination process, but this SARS-CoV-2 virus undergo mutation. Then this spike protein maybe changes. Okay, so when these spike proteins are changes, but the changes are very minimal changes are occurring, not a maximum change. Just like you can see, there looks like this oval shape. Now there looks like the you can say cone shaped. It is not happening. The oval shape structure cannot be changed to the cone shape. Okay, very minimal conversion may be occurring. Very minimal changes may be occurring, and these minimal changes can be accommodated by the particular antibody, because I already said antibody having a different site which is called variable site, and this variable site can change their structure so that they can match with the particular epitope. Okay, if the changes are very minimal, but if the changes are highly variable, it is not minimal, highly variable. In this condition, our vaccines are useless. In this condition, they cannot prevent the entry of virus inside the human body.
Okay, sir. Thank you.
Delta variant.
Okay.
तो एटर mutation হয় নেকি?
কি কৈছা?
Sir, Delta variant যোনটো ওলাইছে এতিয়া
এটো হয় এটা এটা কথা চোৱা বহুত সাইন্টিস্টে কৈছে এতিয়া that delta variant and the delta plus variant, they having the changes in the spike protein, but this protein variation is very minimal. It is not huge variation, very minimal variation. So this variation can be accommodated by the preform antibody already we have. Okay, they can easily they can cope up, but you can say their efficacy may be somewhat less. Suppose the efficacy for the original virus, which is the SARS-CoV-2 virus, the efficacy of the particular Covishield or the Covaxin is almost you can say 80 to 90% efficacy. But it is for the Delta variant, it is for the Delta plus variant, it may be efficacy up to around 70% or 60%. Okay, it may be 20% less, but still they are accurately they can matches with the particular spike protein. Okay, but their efficacy may be somewhat less compared to the original one. Visible? Okay. Okay.
Awesome.
So nowadays before vaccination, before vaccination, because vaccination is done only from the month of January 2021, January or February, it started the vaccination in India only. But before vaccination, one type of monoclonal antibodies already formed by different types of scientists. Okay, and these are called these two. You can see imdevimab and the casirivimab. Imdevimab and the casirivimab, these two are the monoclonal antibody produced only for the spike protein. Okay, by the a group of scientists they produced before the vaccination. When the vaccination process started, now these two are not very useful for the different condition, but they can be useful when if you do not have any vaccination and if you are undergo the infection, then in this condition during the infection phase you can apply this monoclonal antibody to a particular patient. You can apply, but basically they are produced before the vaccination series. Okay, before the month of January or the February. They are started in around suppose in 2020 from September to December. In this area, these particular antibodies are used. Okay, but after that, when the persons are already infected for a particular virus, okay, already infected, they do not go for the vaccination process, they are infected. So in this condition also, they can use these two type of antibodies. These are called monoclonal antibody. Okay, one is the imdevimab and the casirivimab. So these two are the monoclonal antibody. They can derive the IgG G form. Okay, they can derive from the immunoglobulin G form. And these two antibodies are specific for the spike protein of SARS-CoV-2 virus. Okay, and what is the function of these two monoclonal antibody? They just inhibited the attachment of the virus to the human cell. So this attachment is inhibited by the particular antibody. When this virus cannot attach to the human cell, then this virus cannot enter inside the human cell. They cannot enter the human cell. So when they cannot enter the human cell, that means virus cannot infect the human body. If they cannot infect it, after some days, all the virus are dying. in the outside of the human body, and our body is recovered. Okay, if the virus is getting inside the human cell, then what happens? They can proliferate and their lifelong may be extended. Okay, and the serious different types of symptoms may be developed in the human body. Okay, if we prevented the entry of this virus in the human cell, then our body can be prevented from different types of symptoms as well as the different conditions, the morbidity conditions are reduced. Okay, so this is about the definition of a monoclonal antibody, how they are working. Now we go for the history. So how the monoclonal antibodies are produced by different scientists. Okay, you can see in the early 1900, the scientist called Paul Ehrlich. Okay, the scientist Paul Ehrlich, he developed this particular method. Okay, and this method is called as a magic bullet. He called as a magic bullet because one type of drug he developed. This drug can act on a particular disease inside a human body. They can act on a particular disease. Suppose you're having a cancer. Okay, so cancer cells are recognized by the monoclonal antibody. Okay, that means they can specific for a particular antigen. Okay, and this is called magic bullet, and that's why in 1908 the Paul Ehrlich got the Nobel Prize based on a discovery of the magic bullet. Okay, this discovered in early 1900. Okay, and he got Nobel Prize in 1908 in the physiology and medicine. Then you can see in 1975 another two great scientists. One is called George Kohler and Cesar Milstein. So these two scientists, okay, they can produce the monoclonal antibody. They can fuse this monoclonal antibody to the myeloid cell. So myeloid cells are nothing but these are cancer cells. I already said myeloid cells are the cancer cell. So they just generate the monoclonal antibody, and these monoclonal antibodies are fused with the myeloid cell. Okay, and they can form a new type of cell, and this new type of cell is called hybridoma cell. And this hybridoma cell is working as a monoclonal antibody material. Okay, because now question is there, why the myeloid cells are important to produce the hybridoma cells. Okay, now see, when you develop a monoclonal antibody, the particular age period of a monoclonal antibody is very limited. We all know the age of a lymphocyte, how many days it can remain in the blood. For in case of the WBC, how many days they can remain? Almost maybe around 60 days maximum, 60 to 70 days they can remain in the blood. Okay, maximum time. But after 60 to 70 days, all the lymphocytes are died. Okay, so when these lymphocytes are died, how they can produce the monoclonal antibody later? Okay, that's why they can develop a new method. Whatever method they can develop, they first produce the monoclonal antibody, and these monoclonal antibodies are fused with the myeloma cell. Now what is myeloma cell? Myeloma cell is one type of cancerous cell. Okay, cancer cell of plasma cell is a plasma cancerous cell. So this monoclonal antibody fused with the plasma cancerous cell, then they produce a new type of cell which is called hybridoma. Now hybridoma cells are immortal. All the hybridoma cells are immortal. Why? Because these are cancerous cells. We all know cancerous cells are immortal because they can produce and they can proliferate. Okay, by its own, they can proliferate maximum, and all the cells are immortal because these are cancerous cells. So we can extend the life period of monoclonal antibody by attaching this antibody to the myeloid cell. Okay, and based on this technique, they can evolutionize all the medicine industry. They can produce monoclonal antibody for a longer period, and that's why these two scientists get Nobel Prize in 1984. They can get Nobel Prize based on this particular discovery. Okay, 1984 they can get also Nobel Prize. Later on, in 2018, recently in 2018, two another scientists, James P. Ellison and Tasuko Honjo. So these two scientists, they also get Nobel Prize. These two scientists also get Nobel Prize. Why? Because they produce one type of monoclonal antibody which can inhibit the growth of cancer cells. That means these two scientists are working in the cancer cells. So they produce specific type of antibodies which can specific for the cancer cell, and they can kill the cancer cell based on the monoclonal antibody. Okay, and based on this discovery, both the scientists, James P. Allison and Tosuku Honjo. These two scientists also get the Nobel Prize. Okay, so 3 different genera and 3 different scientist groups can get the Nobel Prizes based on the monoclonal antibody. Okay, so this is the glorious history of the monoclonal antibody. Now, the production. How we can produce the monoclonal antibody. Okay, the most interesting part of the production of the monoclonal antibody. Okay, before going to the production, first of all we have to know what is monoclonal antibody and what is polyclonal antibody. I already said, just recall this. Suppose this is a particular virus or maybe bacteria. Okay, so it having certain type of antigen. You can see these are all antigens. Okay, if all these antigens are same, this same antigen can produce same type of antibody. These are all same type of antibody. Okay, and these same type of antibody, they can recognize only one type of antigen. Okay, this is the antigen. This antigen can recognize by this same type of antibodies. Okay, they can recognize, remember, they can recognize only one type of antigen or only one type of epitope. Okay, that's why these antibodies are called monoclonal antibody. Okay, now what is polyclonal? You can see, suppose this is another bacteria or virus. So this is one antigen, this is another antigen, this is another antigen. So 3 different antigens are there. So for 3 different antigen, 3 different types of antibodies are formed. Okay, this is another antibody, this is another antibody, this is another antibody. Okay, so for 3 different types of antigens, 3 different types of antibodies are formed. Now, this group of antigen is not called as a monoclonal antibody. This group of antibodies are called polyclonal antibody because they having the different structures of antibody and they can recognize 3 different types of antigens, not only one type of antigen. Okay, this is the basic difference between the monoclonal and the polyclonal antibody. In case of the natural immunity, we produce this type of immunity. This is a natural condition and this is artificial condition. This is artificial condition and this is natural condition. Okay. Now we go for the most important and interesting part. This is the Hybridoma Technology. Okay. Now what is Hybridoma Technology? For the production of monoclonal antibody. Okay. Now before going to this Hybridoma Technology, first of all we have to know what is multiple myeloma. Okay. So multiple myeloma is a one type of cancer. Okay. This cancer is developed in the plasma cell. You can see multiple myeloma, also called as a plasma cell myeloma or myelomatosis, malignant proliferation of cells within the bone marrow. So inside the bone marrow, the plasma cells are multiple growth occurring inside the bone marrow. Okay, so why they are multiplying inside the bone marrow? Because these are the cancerous cell. That means uncontrolled growth occurs, uncontrolled proliferation occurs of this particular plasma cell. Okay, you can see in the particular diagram. So this is the bone marrow. So inside the bone marrow, this is a normal plasma cell. This is a normal plasma cell. The normal plasma cell can produce normal antibodies. The normal antibodies are produced by the normal plasma cells. If in this plasma cell having the nucleus, if the nucleus is defected or maybe the mutation, if mutation occurs inside the DNA of the plasma cell, then what happens? The uncontrolled growth occurs of this particular cell. Uncontrolled growth. Because the mutation occurs inside the nucleus of the plasma cell. Then they can proliferate numerous type of cell. They can proliferate numerous type of cell. And all these cells can generate a single type of antibody. And this single type of antibodies are called monoclonal antibody. I already said monoclonal antibody can produce by only one type of cell. This type of cell is called multiple myeloma condition. So during the multiple myeloma condition, our own body cell can produce monoclonal antibody. Okay, but this is a disease condition. But normal condition, we cannot produce monoclonal antibody. Now see, in this particular disease, what happened? The disease manifestation as a proliferation of the abnormal plasma cell or plasmablast that populate the bone marrow through the body. They can get it inside the bone marrow, their proliferation is very high. Inside the bone marrow, their development and their maturation is very high. And after some times, they replaces all the natural plasma cells. So these are the natural plasma cells. These are completely replaced by this type of cell, completely replaced. And later on, our body is immunocompromised. Our body is immunocompromised, and our body cannot produce the normal antibodies for the different types of antigens, because this cell only produces a single type of antibody. Okay, suppose we are infected with a bacteria, then our body cannot produce the antibody for the bacteria, because all the cells are becomes infected, these multiple myeloma cells. They can produce only one type of antibody. They cannot produce a multiple type of antibody. That's why our body becomes immunocompromised, and finally we are infected with different types of diseases, and finally death occurs. All the persons are died based on the multiple myeloma disease. Okay, so how we can survive? We can survive by bone marrow transplantation. If bone marrow transplantation occurs, then the patient may be survived. Okay, so this is the disease condition which is called multiple myeloma disease. So these myeloma cells are used in the monoclonal antibody production mechanism, because these cells are immortal. I already said these cells are immortal. If these cells are formed hybrid with a normal monoclonal cell, then what happens? Normal monoclonal cell becomes immortal. And when this normal monoclonal cell becomes immortal, then immortal monoclone can produce numerous antibodies. And these numerous antibodies can use for a different types of treating of diseases. Okay, and they can be stored inside the different storage mechanism for longer period, for years, for thousands of years they can be stored, because all are immortal cells. Okay, so this technology is called hybridoma technology. Okay, now you go for the details of the hybridoma technology. So this is the procedure for the hybridoma technology. You can see. So this is a rat. So the rat is injected by a antigen. I already said we have to inject it only a single type of antigen, because for a single antigen they can produce a single type of antibody, and this antibody is called monoclonal antibody. So the rat is injected with a single type of antigen. This is injected. When the rat is injected by a single type of antigen, inside the rat body they can produce the antibody for this particular antigen. They produce the antibody, and all the antibodies are present in their spleen. In the rat having a spleen cell, in the spleen cell all the antibodies are proliferating and present inside the spleen cell. Then we just collected the spleen cell from the rat, and these spleen cells are go for the purification, and after the purification of the spleen cell we collected the plasma cells or the antibodies which is generated from the antigen. So these are the purified antibodies collected from the spleen cell. Then these purified plasma cells are joined with these tumor cells. These tumor cells are nothing but these are called myeloid cell. These are the myeloid cell. So this myeloid cell and the plasma cells both are unite together. After the union of both the cells, they can produce a new type of cell, and this new type of cell is called hybridoma cell. Okay, and these hybridoma cells are immortal, and they can produce monoclonal antibodies for longer period, for longer time period. Okay, and finally these monoclonal antibodies are purified, and these are used for a particular disease, for a particular antigen. Suppose this antigen is for the spike protein of the virus, of the coronavirus spike protein, this is the antigen. Then these antibodies are the specific for the spike protein only. They can bind to the spike protein of the particular virus, and they are called as the monoclonal antibodies. Okay, so this is the normal detailed technology, but this technology is somewhat longer because some other steps are also there. Now we discuss the other steps related to the hybridoma technology. Okay, so these are the basic steps of the hybridoma technology. Okay, we go for the first step. You can see identification of plasma cell for specific antigens to produce monoclonal antibody. First, we have to identify the particular antigen so that we can produce the antibody. Suppose we are talking about the this one, spike protein for the coronavirus. So we have to collect the spike protein first, and these spike proteins are injected to the this one rat. We injected the spike protein to the rat. Then inside the rat what happen? They can produce the monoclonal antibody inside the rat. Okay, and these monoclonal antibodies are collected from the spleen cell of the rat. We collected from the spleen cell of the rat, and they are purified. And after purification, we get the all monoclonal antibody which is generated inside the rat. Okay, then what happened? The next step is that all these cells, all the monoclonal cells, they are fused with the myeloid cells. They are fused with the myeloid cell with the help of a particular material which is called polyethylene glycol. This is a material called polyethylene glycol. This is a material. This material is used for the fusion of both the cells. Both the cells are fused together when we apply the polyethylene glycol. Okay? After application of this, they produce a new type of cell. This is called rat hybridoma cell. They can produce the rat hybridoma cell. Then, the myeloid cells have lost the ability to synthesize hypoxanthine, guanine, phosphorybacil transferase. So there is an enzyme. There is an enzyme called AGPRT. The enzyme called AGPRT, hypoxanthine, guanine, phosphorybacil transferase. So this enzyme is lacking in the myeloid cell. Myeloid cell cannot produce this particular enzyme because myeloid cell is the cancerous cell. So this cancerous cell, they cannot produce this particular enzyme. Okay. So why this enzyme is important? Because this enzyme is necessary for the synthesis of nucleic acid. For the synthesis of the nucleic acid, this enzyme is very much essential for the myeloid cell. Okay. So if you collect the myeloid cell and they are stored in the outside of the human body, all the myeloid cells are died within some days because they cannot synthesize this particular enzyme. If they cannot synthesize this enzyme, myeloid cells are died because they cannot synthesize the nucleic acids. That means they cannot proliferate. If they cannot proliferate, all the myeloid cells are died after some days. But they can also survive by another mechanism. The another mechanism is called this one. The absence of HGPRT is not a problem for the cells. It is not a problem for the myeloid cells because they having another mechanism. This mechanism is called de novo purine synthesis pathway. They having a de novo purine synthesis pathway. It is also one type of pathway which can also generate the nucleic acids by another synthesizing pathway. That means myeloid cells, they cannot synthesize the AGPRT enzyme. That's why they cannot synthesize the nucleic acid. But they having an alternative mechanism to synthesize their nucleic acid. And this alternative mechanism is called de novo purine synthesis pathway. If we can able to block this pathway, if we can able to block the de novo protein synthesis pathway, then all the myeloma cells are died because they do not have this enzyme. If you block this pathway also, then all the myeloma cells are died. That means they can survive by this alternative mechanism. Okay. Now, how we can block this pathway? How we can block? We can block this pathway by a particular molecule. And this molecule is called this one, aminopterin. This molecule is called aminopterin. Okay. Suppose we have collected the myeloma cells, then we have applied this chemical, the chemical called aminopterin. So this chemical is an analog of this particular molecule. This molecule is called folic acid. So aminopterin is an analog for folic acid because it can inhibit the dihydrofolate reductase. There is an enzyme called dihydrofolate reductase. Is an enzyme. So this enzyme is inhibited by this aminopterin. Aminopterin can inhibit this particular enzyme called dihydrofolate reductase. If this enzyme is inhibited, then the myeloid cell cannot synthesize the what? DNA molecule, nucleic acid. They cannot synthesize. And finally, all the myeloid cells are died within some days because they cannot synthesize nucleic acid by the de novo protein synthesis pathway because this pathway is blocked by the aminopterin. Okay. So if you going to treat the myeloma disease condition, we can apply the aminopterin molecule. And imidatorin molecule, they can bind with the dihydrofolate reductase enzyme, and then they cannot synthesize this particular enzyme, or all these enzymes are blocked by the entry of the aminopterin, and they cannot synthesize the nucleic acid molecules. And finally, all the myeloid cells are died within some days. Okay, so this alternative pathway can be blocked by the aminopterin molecule. Okay, so question is that why this pathway is needed? Why you need to apply the aminopterin? Okay, this is needed that during the hybridoma technique, we do not know which cells are hybridized and which cells are not hybridized. We do not know. Okay, so how we can consider those cells are hybridization molecules and those cells are non-hybridization molecules? How you can consider? We can consider by the use of this particular technology. By the use of this particular technology, we can collect only the hybridoma cells from the non-hybridoma cells. That is, you can also say the selection procedure. You can also say the selection procedure of the hybridoma cells. Okay, so how we can select? We can select by a particular medium, and this medium is called hat medium. You can see hat medium. The selective culture medium is called hat medium. Now what is hat medium? Hat medium having the hypoxanthine, aminopterin, thymidine. Hat means hypoxanthine, aminopterin, thymidine medium. So this medium is applied for the hybridoma technique. You can go for the next, first go for the earlier diagram. Okay, now see. So we are injecting the particular mice, they having the spleen cell can produce the plasma cell, and all the plasma cells are purified from the spleen cell. Then they are binding with the myeloma cells and they can produce the hybridoma cells. Okay, so during this technique, these hybridoma cells are hybridized or not. How we can define? Okay, they do not may be hybridized. They may be remain as a plasma cell. They may be remain as a myeloid cell. Okay, we do not know which cells are hybridized. So we have to select those cells are hybridized. These are essential. These cells are not essential. These cells are not essential. We need only the hybridized cells. So how we can select? For a selection procedure, we are applying the hat medium, which is called hat medium means what? This one. Hypoxanthine, aminopterin, thymidine medium. So this medium is applied. Okay, and in this medium, all the hybridoma cells are put. All the hybridoma cells are put in this particular medium. Now, what is happening in the hat medium? Now see, in the hat medium, aminopterin molecule is there. In the hat medium, aminopterin molecule is there. What is the function of aminopterin? They can block the de novo synthesis mechanism of the tumor cells, because tumor cells do not have the HGPRT enzyme. They cannot synthesize the what? They cannot synthesize the nucleic acid by HGPRT enzyme. They cannot synthesize, but they become alternative mechanism. The alternative mechanism is called de novo purine synthesis mechanism. Okay, and this mechanism is blocked by only one molecule. This molecule is called aminopterin. So in the hat medium, the aminopterin molecule is here. Okay, because of the presence of the aminopterin molecule in the hat medium, this aminopterin molecule, they are getting inside these cells. All the non-hybridoma tumor cells, they are getting inside the aminopterin molecule, and then they can block the synthesis of amino acid, synthesis of the nucleic acid inside this particular cell. They block. After blocking of this, all the tumor cells which are non-hybridized, they are died. This is one condition. Next condition. Suppose only the plasma cells are there in this hat medium. So plasma cells having a normal life period. What is the life period of plasma cells? Only maybe for up to 60 to 70 days they can life condition. So all the hybridoma cells, they are remain inside the hat medium for 60 to 70 days. Okay. After 60 to 70 days, all the plasma cells which are non-hybridized, they are died. Okay, because in normal condition, they all are died. But after 60 to 70 days, those cells are survived in the hat medium. That means those cells are hybridoma cells, because these cells are immortal, because this cell having the fusion of this cell and this cell. Okay, the hat medium aminopterin molecule cannot affect these cells because this plasma cell having the normal mechanism of HGPRT mechanism. These cells having the normal HGPRT mechanism. And the HGPRT mechanism, they can synthesize the DNA molecule. They can synthesize. So because these cells are binding with these cells, they have a normal mechanism. That's why all the hybridoma cells, they are alive in the head medium. But those cells which are not hybridized, only the tumor cells, they are died because in the head medium, aminopterin molecule is there which can block the de novo synthesis of the purine molecule. And finally, we get only the hybridoma cells in the head medium. And these cells are purified, and for the purification of the cells, we get the monoclonal antibody later on. Okay? So this is the technology, and this technology is called Hybridoma Technology. Okay? Now, the next important parameter. So after the screening of the Hybridoma Technology, how we can know, how we can screen all the hybridoma cells, how you can screen. So we can screen the hybridoma cells by two techniques, by ELISA technique or by dot blot technique. Immunoblot or a dot blot. Immunoblot technique or the dot blot technique or by the ELISA technique. We can select the hybridoma cells. So how we can select? Now see, all the hybridoma cells, they can produce antibody for a specific antigen. For a specific antigen, they can produce the antibody. Now see, suppose we are producing the antibodies for the spike protein. That means these antibodies are specific for the spike protein. Now, how we can select they can produce the antibody for the spike protein or not? So in this condition, we are applying the ELISA technique. Okay, in the ELISA technique, in the ELISA plate, we just put the spike protein in the ELISA plate. In the ELISA plate, first we put the spike proteins. Then we applied the monoclonal antibody over here, which is already prepared. If the monoclonal antibody can bind with the spike protein in the ELISA plate, that means your hybridoma technique is successful because all the spike proteins are binding by the antibodies which are produced by the hybridoma technique. They can bind. If they can bind, that means your technique is successful. If they cannot bind, that means your hybridoma technique is completely failed because they cannot bind the spike protein in the ELISA plate. Okay, so we can select all the monoclonal antibody based on the ELISA plate technique. Okay, now see, the most productive and stable clone then selected for the future use. So those are the most stable clones, they are used or stored for the future use. Okay, then see, they can be also injected into the mice. Okay, so when you produce the monoclonal antibody, this monoclonal antibody again can be injected in the mice. Okay, so when you can inject it into the mice, what happens? Inside the body of the mice, they can produce a particular fluid, and this fluid is called ascitic fluid. They can produce the ascitic fluid inside the body of the mice. They can produce the ascitic fluid. And what is the ascitic fluid? Inside the ascitic fluid, all the monoclonal antibodies are proliferated maximum. All the monoclonal antibodies are proliferated maximum inside the ascitic fluid. That means we can produce the monoclonal antibody inside the mice up to a maximum use. We can produce inside the mice by proliferating the hybridoma cells inside the mice, inside the ascitic fluid. And later on, we can collect the ascitic fluid, and these fluids can be purified for the hybridoma technique. We can purify. This is a technique. But this technique is nowadays, it is completely banned. Why it is banned? Because this is unethical. You can see whether ascitic formation is an unethical process. Because in this condition, the mice or the rat can be killed. It can be killed to collect the ascitic fluid. When we collect the ascitic fluid, all the mice are killed. So if you kill all the mice, that means it's one type of unethical condition. That's why this particular mechanism is now stopped. Okay, now we are using an alternative mechanism, and this alternative mechanism is called recombinant DNA technology. Now we are using the recombinant DNA technology for the production of monoclonal antibody inside the viruses and inside the bacteria. Inside bacteria, inside the viruses, we can produce the monoclonal antibody, and this is called recombinant DNA technology. Okay, and this mechanism is completely stopped because it is unethical, and all the mice have to be killed to collect the ascitic fluid. Okay, so you can see this is the injection of this particular mice, and from the ascitic fluid we can collect the antibody. This is one mechanism, or otherwise we can get the antibody from this particular mechanism. Okay, now the most important part I already said. The next one, the development of the monoclonal antibody by recombinant DNA technology, or maybe called as a phase display. This is called phase display mechanism. By phase display mechanism, we can also produce the recombinant monoclonal antibody inside the phase virus. Inside phase virus, that's why called phase display. Bacteriophage. So inside the bacteriophage, we can produce the monoclonal antibody. So how we can produce? You can see the mechanism. You can see a simple method of construction of human monoclonal antibodies can be accomplished using the recombinant DNA technology. By using the recombinant DNA technology, you can produce the monoclonal antibody. Okay, you can see once a mouse monoclonal antibody has been constructed, first of all we have to construct the monoclonal antibody inside the mouse, then just describe DNA encoding the antigen binding portion of the antibody molecule can be isolated and fused to human DNA that encode an antibody. Okay, now see, first we have to gather the monoclonal antibody from the mice. Okay, then we isolate the particular gene, the gene which can produce the monoclonal antibody inside a particular monoclonal cell. Okay, first we have collect the plasma cells, hybridoma cells from the mice, then we can collect the gene, specific gene which can produce the monoclonal antibody inside the hybridoma cell. Then this gene is cut down, and this gene is inserted into the particular DNA of the human DNA, which human DNA also encode for the antibodies. Suppose a human DNA which is also encode a antibody. Okay, so in this human DNA we can insert this particular DNA collected from the hybridoma cell. Then we get a new DNA, hybrid DNA, and this new hybrid DNA is inserted to a particular bacteriophage, and this bacteriophage is called lambda phase. So in the lambda phase, we are inserted this particular modified gene. The modified gene having a segment of a human DNA which can produce the antibody, and a segment of DNA which can produce the hybridoma monoclonal antibody. And both the gene segments are introduced inside a phase virus, inside a lambda phase virus. Then the lambda phase virus now can produce the monoclonal antibody inside their body. This is called phase display. What is phase display? When they having the particular gene segment inside the phase virus, then what happens? The phase virus can produce the monoclonal antibody in their body. In their body, they can produce the monoclonal antibody. And this condition of production of monoclonal antibody in their body is called the phase display. Okay, now see. So how we can select which virus having the phase display or not? Because all the viruses are there, we have to select the only one virus which having the phase display. Okay, so how we can select? We can select by the same process of the ELISA technique. The same process of the ELISA technique we can select. You can see. So in the ELISA technique, we can get the particular antigen. Suppose this is the spike protein. This is spike protein antigen for the virus. Okay, for the virus of SARS-CoV-2, this is a spike protein. It is attached to the ELISA plate. Then we can apply all the viruses, bacteriophage lambda, applied in this particular well. Then what happened? Those having the displaying protein, this is a monoclonal antibody which is displayed, blue color, but these are not displaying protein. And this blue color display protein, they can bind to this one antigen, they can bind. But these viruses cannot bind. So this binded virus is the desired virus, because this virus having this spike protein, this monoclonal antibody protein. Okay, then later on we will go for the washing technique. By washing technique, all the other phase viruses are washed out. and only the phage virus which is binding to the antigen, they are remain there. Okay, and then we selected these viruses, and these viruses are applied to proliferate inside the bacteriophage. That means bacteriophage are attacking the E. coli virus. Now we are applying these phage viruses into the E. coli bacteria so that they can proliferate into maximum viruses, and they can proliferate into maximum viruses inside the E. coli bacteria. And later on, we collected all the viruses, and these viruses are fragmented, and we can collect the specific type of monoclonal antibody from this virus because it is already prepared inside the particular virus, already prepared. But we have to proliferate. How we can proliferate? We can proliferate this specific virus inside the E. coli bacteria. So we have to use this lambda phase inside the E. coli bacteria. And inside the E. coli bacteria, they can proliferate to maximum number. When they becomes maximum number, we have to collect all the phage viruses. We have to collect. Then, by the fragmentation process, we collected only the monoclonal antibody from these phage viruses, and it is applied for the future use. Okay, so this is a new type of technology, and this technology is extensively used for the production of the monoclonal antibody nowadays, and this technology is called recombinant DNA technology. But other one is the non-recombinant technology, where the mice are sacrificed by ascitic fluid, so that after the sacrifice of the mice, we get the monoclonal antibody from the mice. So this procedure is nowadays stopped, and we can apply this particular modified procedure for the collection of the monoclonal antibody. And this is a widely used technology called as a recombinant DNA technology. Okay, so this is about the normal development or the process of the recombinant DNA technology to form monoclonal antibodies. Now go for the next one, application, where the monoclonal antibodies are applied. Okay, most applications are basically, these are the applications you can see. Number one, diagnostic test. Okay, so to detect the antigens, in the human body. Suppose we are infected with the SARS-CoV-2 viruses. So in the human body, the SARS-CoV-2 antigens are there. So how we are detecting we having the SARS-CoV-2 disease or not? How we can detecting? So for this condition, we can apply the monoclonal antibody. You can apply it. By applying the monoclonal antibody inside of a human body, we can detect our body is having SARS-CoV-2 virus is present or not. Suppose the technique is called this one, immunohistochemistry. Immunohistochemistry is a particular technique. So in this technique, we can detect the different types of antigens. So suppose we have to detect a particular antigen for a particular virus. Suppose we are infected with a particular virus. We do not know whatever virus it is. Then we just collected our body cells. And then the body cells are undergo a particular technique called immunohistochemistry. And by this technique, we are applying the antibodies into a particular cell. In a particular cell, we apply the antibodies specific for the particular antigen. Then what happens? All the antibodies are bound over the antigen which is present inside the body cell. That means we can confirm that our body cell is infected with a particular virus. We can confirm. Suppose we are infected with the SARS-CoV-2 virus. If it is present in the body cell, we have to collect the body cell, then undergo immunohistochemistry, and inside the immunohistochemistry, we are applying the specific monoclonal antibody for the SARS-CoV-2 antigen. Then when we apply the monoclonal antibody to the body cell of the immunohistochemistry, then what happens? They can bind easily to the antigen. When they can bind easily to the antigen, then we can say that our body is infected with the SARS-CoV-2 virus. Okay, and this is called diagnostic test. But this test is very costly, because in this test we have to identify the monoclonal antibody first. And it's a very tedious process. You can all know that it's a very long process to develop the monoclonal antibody. Okay, so this diagnostic test is very costly for applying the monoclonal antibody. The next one, analytic and chemical uses. Okay, we can also analyze the different epitopes or different targets by the help of this monoclonal antibody. Same that of this diagnostic test you can also use. Okay, the second time is the analytic or the chemical use, to use the different types of chemicals as well as to detect different types of epitopes or the antigens. We can apply the analytic purpose. So this purpose is also costly because it is also known as monoclonal antibody. The next one, therapeutic use. In the therapeutic use, it is also used. You can see therapeutic monoclonal antibodies act through multiple mechanisms such as blocking targeted molecule functions, including apoptosis in the cells which express the target and by modulating signaling pathway. Okay, now see, suppose your body cell undergo apoptosis. Now what is apoptosis? Cell death, which is called programmed cell death. In some condition our body cells are dead automatically. Okay, or some type of body cells are dead due to some abnormal condition. All the body cells are dead, they are died. So when the cells are died, that means you have a particular disease. In this disease condition, all the body cells are somewhat trying to die. So to prevent this particular mechanism or the cell death or the apoptosis, we can apply the monoclonal antibody. So we can detect a particular enzyme or a particular epitope, okay, of this particular cell death mechanism. Then from this epitope, we can generate the monoclonal antibody. Okay, so when they generate the monoclonal antibody and this monoclonal antibody can be applied to the particular person, then what happens? Inside the person, the cell death mechanism, apoptosis mechanism can be blocked by the monoclonal antibody because they search for the particular antigen which can generate the cell death mechanism. If this antigen is searched by the monoclonal antibody, then they can inhibit the mechanism of apoptosis process, and that's why we can inhibit the mechanism of apoptosis inside the cell body, and then our body cells can be revived again. Okay, so this is a one type of therapeutic use. Next one, cancer treatment. So in the cancer also we can use the monoclonal antibody. I already said two scientists, they get the Nobel Prize in 2018. Ellison and the Hanjo and the Ellison, these two scientists getting the Nobel Prize based on the cancer treatment. So for the cancer treatment, monoclonal antibodies can be used. Okay, the last one, autoimmune disease. So in case of some autoimmune disease also, monoclonal antibodies are used. The autoimmune disease like this one, rheumatic arthritis, or maybe the Crohn's disease, yes. In the Crohn's disease and the rheumatic arthritis, we can use the autoimmune. This type of autoimmune disease, we can use the monoclonal antibodies. So monoclonal antibodies are just like this one, infliximab and the adalimumab. So these two are the monoclonal antibody. They can be used for the rheumatic arthritis and the Crohn's disease. Okay, so these are the applications of the monoclonal antibody. So these are the 5 different types of applications of the monoclonal antibodies. Okay, so this.
So diagnostic test to ako ebar bujai dibo paribo.
Okay, so diagnostic test means suppose just I am giving an example of the SARS-CoV-2 virus. Okay, suppose you are infected with the SARS-CoV-2 virus, then what happens? Your body cells having the antigen, the spike protein, you having the spike protein in your whole body. Okay, then how we can detect you having the SARS-CoV-2 infection or not? So in this condition, you have to collect your body cells. First you collect your body cells, then in the body cells already having the spike protein, we all know. Now we can generate the monoclonal antibody for the spike protein. We already generated. Okay, then we apply the monoclonal antibody upon the collected cells from your body. Okay, in the collected cells from your body, we apply the monoclonal antibody. Then what happens? If in your cells having the spike protein, then the monoclonal antibody, they can bind over the spike protein. If they are binding, that means you can detect that you are infected with the SARS-CoV-2 virus. This is a detection process, and this is called diagnostic test. Okay, but this test is very costly because in this condition, first we have to generate the monoclonal antibody to detect the particular antigen. Okay, but we having an alternative mechanism. We all know rapid test. We having the very easy process, rapid test. By using the rapid test, we can diagnose the coronavirus detection in our body. This is very effective and less costly. But this is also a detective mechanism, but this mechanism is very costly because first of all we have to generate the monoclonal antibody, then you can detect the particular antigen in the body. So busy pala kotha to idea?
Okay, sir.
Okay. So this is about the different types of application of monoclonal antibody. Okay, so this is about it. So these are some references, reading materials, and then thank you.